just pipette the reagents on top, and normally they take them relatively easy. But when we talk about treatments or using CRISPR in vivo, it's a bit of a different story, because if you think, for example, of a disease in the liver, that you have to target cells in the liver, the organ has so many cells and so many different types of cells. How do you make sure that you are targeting the right cells and in a sufficient amount so that you can actually revert and cure the disease? So this is still a little bit of an unknown field. A lot of research is going into this, into different methods to deliver the CRISPR reagents, so the therapeutics. But currently, not all the organs in the body are possible to target with CRISPR. The liver is one that is possible to do it with lipid nanoparticles. This is a very easy, basic cartoon just to recap on what I've just told you. So you have the components of the CRISPR system, the Cas9 that is complexed with a guide RNA. This guide RNA contains two parts, so the CRISPR RNA and the tracer that together form the single guide RNA. And it's the CRISPR RNA that is a sequence of 20 nucleotides that is going to determine where the Cas9 is going to cut the DNA. And why? Because these 20 nucleotides have to be complementary to the sequence of the DNA that we are interested to target. And that only happens when there is a PAM site just before. So these 3 nucleotides, in the case of SpCas9, that is the beginning of the CRISPR edits. So the complex of Cas9 and guide RNA is going to scan the whole DNA to find PAM sites. Whenever there is an NGG in the DNA, the whole Cas9 is going to unwind the DNA and open it in order to check if the full sequence, the 20 nucleotides of the CRISPR RNA, are going to be complementary to the 20 nucleotides that are after the PAM. If that is not the case, the whole system closes again, and the CRISPR-Cas is going to scan until it finds another NGG PAM site, opens the DNA, checks for the complementary of the guide, and if that's the case, then a double-strand break is going to be introduced after 3 nucleotides after the PAM. Is it clear? Yeah. Yes?
So the whole complex, in case there's no complementary, they do it again. So does that mean that there are more than one PAM site?
There are, in the genome, there are thousands of NGG sequences. So that is going to determine where CRISPR can work. If there is no PAM site, the system will just continue. That's how it works. And then what determines this specificity is then after the PAM is found, is the sequence of the 20 nucleotides that come after, and that is determined by the sequence of the guide RNA that we design, that we provide the Cas9 with, so that it can scan and find the targets. Yes?
And how is the guide RNA produced? Is it produced by us in the lab? Or?
Yes.
Okay.
It's
so
produced in...
Do we make it according to the DNA so we know that it's complementary?
Exactly. So nowadays, the whole genome is, human genome is sequenced, and multiple times with in multiple individuals, and so there is a sort of annotated whole genome sequence, and based on that, we can now use that information to design sequences of 20 nucleotides that can be used as a guides for this kind of CRISPR activity.
Is it then patient-specific or just human?
So in the case of patients, there are mutations associated with the diseases, and those are annotated per gene. In the whole genome, there are other variants that are small variants that in between people exist, but those are not causative of disease. Those are also annotated in this whole genome database. Yes?
Does it happen that there's no NGG sequence close to the gene that's being targeted, or is the
Sometimes it's difficult to find a good design for the CRISPR guide, let's say, but NGGs exist everywhere. Yeah, I should know the number by heart. I knew it once, but I don't remember anymore how many NGGs we have. Okay, if it is clear, we continue. So this is all very nice, but making this double strand break is just half of the job. Actually, it's up to the cell to do the rest of the work in order to have edits. Do you know what I'm talking about? Yes.
Or incorporate modification and fix the double strand break.
Exactly. So the DNA is the only molecule in the cell that is not replaceable. You only have that is just one, and the cell protects this molecule with lots of care. And so having a double strand break in the DNA is not good, and the cell will immediately try to repair that through mechanisms that the cell has in place that are called DNA damage response. And there are several of them, but for CRISPR and for today, we'll be focusing two of them. And one of them is called non-homologous end joining. And as the name kind of suggests, after a double strand break, the cell is going to try to glue the two parts together in order to correct that as fast as possible. This is a mechanism that is present in the cells throughout the cell cycle, so it's always in place. Because it's very fast, it also has the disadvantage of being very error prone. So when cells start to glue this immediately, there are chances that some nucleotides are lost, some nucleotides are inserted, and so what you have at the end of this non-homologous end joining mechanism is actually a gene that is disrupted because of some nucleotides that were inserted in this gluing process and others that was lost. And so it is in this case that you can inactivate genes with CRISPR. It's actually when the cells try to repair the double strand break and use this method that is very error prone. In this case, and in the context of CRISPR, there is another term that you find it in the literature everywhere, that is indels. And indels is the abbreviation for insertions or deletions that are the result of this non-homologous end joining that at the end introduces alterations in the sequence of the gene. And this lead to frame shift mutations, for example, and so the information that the gene contained originally is now altered because of 1, 2, 3, 4 nucleotides that are incorporated there. And you know that the DNA is read per 3 codons, and so if you introduce one nucleotide extra or delete that, you will have a completely different message when you read that gene afterwards. So this is again what we use when we want to inactivate genes or generate knockouts. It's also another term for that. And how do we know that the genes were inactivated afterwards? Because then we are going to look into the sequence of the DNA, in the target sequence where we know where the guide is going to bind, and we can sequence that fragment to understand what are now the nucleotides that were inserted or deleted, or what actually is the final product of this CRISPR experiment. And by sequencing, you can determine the sequence of nucleotides that you find. And this is just a way to represent it. So here you see that this 0 indicates the wild type sequence. And then if you have +1 or -1, -2, -3, etc., you have different percentage of the indel that is found in the whole cell population that received the CRISPR treatment. Yes?
How can we make sure the cell uses this type of DNA repair and not another type of DNA?
So normally the cell is going to use this one because it's the one that